Okay, so this is AQA Paper 1 2018. Now I'm starting on question 1 as I'm working my way through the paper, but I'm going to do something a bit weird here. I've just recorded all my exam technique videos, and my advice to you starting Paper 1 for AQA is to go straight to the final question. The final question is well, look at this. We've got a full size of A4. It's a 6-mark question. This is what tells you how much you need to write. It's not how much space is available. We have typically three questions totaling 15 marks. This is the format of the paper. These are described. This is an AO1 question. So assessment objective 1, tell me what you know. Compare and contrast is assessment objective 1, 5 marks, and the growth structure of the human gut. So this is described. So these are 3 tell me what you know. These are the questions that students all the time say that they find the easiest. There's not really much mystery with these questions. AQA Paper 1, your exam technique is start with the final question. Also, a little note to self, 41 pages. This exam paper is 42 pages long. It's an insane amount of space. If you try and fill the answer space for every question, getting up to question 10, you won't get here. And these are the easiest questions on the paper. There's 15 marks, it's whatever, it's out of 91, it's 16.5% of the paper here. And you can probably smash out your answer pretty quickly if you know these topics. If you don't know them, you don't have to do them now. But if you know these topics, then you're going to basically know you're going to get 16% of the paper in the bag from the beginning. The rest, well, there's another 10% of the paper is going to involve math skills, which if you've practiced lots and lots of, you're going to get pretty much full marks on all those maths questions. So that's 26% of the paper. And then 15% of the paper's on practicals. If you've researched them really well, then you're probably going to get, most of those marks. That puts you on, you're probably getting a C already, and you haven't even had to think really very much outside of a very limited part of the specification. So watch my exam technique videos. I've done thousands of these things. I know what I'm talking about. So follow my advice and you are definitely going to improve. So my rant over on that, but start with that final question. Do not leave those final questions unanswered, because otherwise you're just not answering the easiest, most accessible marks on the paper. Into question 1. Figure 1 shows all the chromosomes present in one human cell during mitosis. A scientist stained and photographed the chromosomes. Okay, so they have been stained, otherwise you can't see them. Any chromosome that you can see has been stained. Figure 2, the scientist has arranged the images of these chromosomes in homologous pairs. Okay, so we have our sex chromosomes, X and Y. Y chromosome being a lot smaller than the X chromosome, hence why we get lots of sex-linked characteristics, and then the other 22 pairs. Give is our command word, so that's an AO1 command word. Just tell me facts. 2 pieces of evidence from figure 1 that this cell was undergoing mitosis. Explain your answers. Okay, so it's not clear by any means, but maybe I would recommend you printing these papers out and actually doing them in the flesh. What are these little tiny little bits here? Here. I mean, that's one bit of evidence. What do cells do before they do mitosis? Anything from figure 1 we're using, so we're not using figure 2 here. Well, the chromosomes also are, okay. We've got this little notch here, which is unusual if the cell, and we've also got the fact that the chromosomes themselves are visible, which ordinarily they wouldn't be. What else can we say? I think that's probably our 2 things that we can say to get those marks there. Okay, so we've basically got a tick box. Which box is the name of the stage of mitosis? So it's the stage of mitosis, which is shown in figure 1. So again, just making sure we know which one we're looking at. Anaphase, interphase, prophase, telophase. You're going to be expected to know those, whether that's from a diagram or whether that's producing 2 bullet points to describe each of these phases, or to be able to recognize it in a micrograph like this. When preparing the cells for observation, the scientists placed them in a solution that was slightly higher, less negative water potential than the cytoplasm. So slightly, I think, less negative. I mean, the terminology here is always a bit confusing, than the cytoplasm. This did not cause the cells to burst, but move the chromosomes further apart in order to reduce the overlapping in the chromosomes when observed with an optical microscope. Suggest how this procedure moved the chromosomes apart for 2 marks. Okay, so it's not going to burst I mean, I don't think that's relevant other than the fact that we don't need to talk about it bursting or because they've told us it hasn't. Why, imagine a balloon, if you're drawing a chromosome or a face on a balloon and it's like 3/4 full, if you then add some extra air to the balloon, what happens to the distance between the smile or the distance between the eyes as the balloon gets bigger? Well, that's basically what you're doing here, and you just need to translate that principle into osmosis and the size of the cell and link it all together. So question 1 continues. The dark stain used on the chromosome binds more to some areas of the chromosomes than others, giving chromosomes a striped appearance. Suggest one way in which the structure of a chromosome could differ along its length to result in the stain binding more in some areas. Okay, so this is about chromosomes and their makeup. So what are chromosomes made of? They are densely folded bits of DNA, the polymer. And what are they folded around? That might point us in the right direction. I don't know the answer. This isn't, it's a suggest question. It's not something that is supposed to be on the spec. It's not in the course in a specific bullet point that you can go and find, but it is, you're going to have to apply your knowledge and figure out what do we know about the structure of chromosomes? When you get to questions like this, what do you know about the structure of chromosomes? Well, not a huge amount, because the course doesn't go into it in a huge amount, but you do know a few bits, and so more than likely this is going to be linked to that. In figure 2, again, one of the things, a skill that you really have to be practicing is working on a question where you cannot actually see the question. So this is figure 2, and you can't see the figure 2 whilst we're reading the question. The chromosomes are arranged in homologous pairs. What is a homologous pair of chromosomes? Okay, so we've got 1 mark. What are the features? What genes do they carry? What can you tell me? Again, this is a great one for carbs, so command word and marks. We've got what is our command word. We've got 1 mark. What's the actual topic of the question? Well, homologous pairs. There's not too much mystery to this one. Can we rephrase the question? Yes, I think we can. We can say, what are the defining features of a homologous pair? I think, what do you know about them? There's not that much you have to know about them for 1 mark. Again, we've got give, so it's a straight up, tell me what you know. Give 2 ways in which the arrangement of prokaryotic DNA is different from the arrangement of human DNA. Okay, so we can just basically swap this out for eukaryote because eukaryotic DNA is arranged in the same way. So 2 ways that DNA is different in prokaryotes than it is in eukaryotes. And again, there's a bit of a directionality here. It's asking us from how is prokaryote different from eukaryote, although I think we can probably say this one has this feature, whereas the other one does not. It should be fairly easy for if you know your facts.
To get more biology videos from me, subscribe to the channel, and of course don't forget to try out my complete A-level course in the free trial over on the Tailored Tutors website. Up next, you can transform your grade in about an hour with my grade transformations playlist, or you can watch the video that's been magically selected for you from the people at YouTube.